5 years from now, I think we're going to be taking a molecule that's basically ready for prime time directly to a patient, potentially. I don't think we're going to be quite doing zero-shot clinical design, like literally taking that molecule straight to a patient yet in 5 years. But I think you'll see molecules that effectively are basically good enough for that. Because, and part of that I think about Manifold, we already have molecules that we put in vivo and they look really, really good already.
So you're saying like first in human in less than 12 months kind of thing.
Yeah, exactly.
Gleb, thanks for joining us on Transcribed.
Thanks for having me.
Fun fact, I was recently reminded that you were my TA way back in undergrad when I was a freshman in 601. So we've known each other for a while, and we'll definitely come back to your roots in computer science. But let's start with Manifold first. Can you tell us a little bit about Manifold Bio?
Yeah, of course. So Manifold Bio, we are a platform therapeutics company. So we have created a way to generate a very, very high scale of data at the in vivo stage of drug discovery. So this fundamentally challenges the paradigm of how drug discovery can be done. When you can ask some of the hardest questions early on in the process, you can take on very hard problems that have not been solved before. So there's two parts of the company. There's the AI design side of things. So we create our own antibodies and other molecules. And then there's this molecular barcoding technology that lets us generate this really high scale in vivo data. And then we put that together and we create drugs and are quite focused on drugs that are tissue targeted, meaning they go to specific places in the body.
So like in a pre-manifold world, like how does in vivo, how do in vivo studies work kind of pre-manifold technology?
Yeah, so the classic thing in this world, we always talk about funnels. And the way that drug discovery is done is there's a funnel at the top where you're testing a lot of things.
Sure.
At the bottom, there's basically the one drug that's going to go to patients. So classically, at the very top, you would start testing a lot of different drug designs. You do those tests directly on cells. Those cells are typically in a petri dish. This is the in vitro paradigm. And you can take and automate those steps so you can get a lot of scale. And even today, a lot of folks are talking about the power of automation to scale up those tests. But fundamentally, a petri dish is not a person or even a living being. And so to really understand what happens when you bring a drug to a living being, you end up testing it in an animal. And you want to see whether it's safe, whether it's starting to do the right thing. That step could only be done a few at a time. Just you're testing one drug per animal, so you want to use as few animals as possible. And so you have this funnel that kind of looks like thousands or tens of thousands of molecules at the top, like on the order of tens in a mouse, maybe a couple in a monkey, and then you're advancing to humans.
Yeah, so sort of scaling in vivo testing today, kind of pre-manifold, looks like more in vivo techs and more mice, right?
That's
It's
right.
like very, very difficult to scale.
That's right.
And very low throughput, it sounds like.
Yeah, it's kind of one of those things that even if you're kind of the most even if you have a ton of money, you still are going to hit the limits of you don't really want to use a ton of animals for this step. From an ethical perspective. So it's a bit of it's classically the kind of bottleneck that feels or felt like it would not be possible to break through.
So give us a sense of scale. With Manifold's core technology, how many antibodies could you screen in a given animal, and how big of a leap is that from the traditional method? What's now open to us that wasn't before, especially when it comes to you have AI now generating proteins that you want to test in these animals?
Yep. Yeah, and so actually the AI piece is really interesting. You could have, today you can generate millions of candidates. You can have all these amazing ideas for drugs, but you still can only test a couple in the most important in vivo systems. The scale that Manifold can now do is we are regularly doing over 100,000 antibody designs per single animal study. And in fact, we recently did a study with over 500,000, actually it was 587,000 distinct antibody designs that went into a single
One mouse?
NHP. No, NHP.
NHP, wow.
Monkey. And so that was, again, like really want to reserve that stage for something that is really, really typically mature, and you've already invested a lot, and you want to make sure that that's a good use of that stage.
I mean, NHP studies are really expensive. I know companies that spend like $1 million per NHP study.
That's right. So now with this scale, this fundamentally changes everything for how we're doing things.
That's so cool.
Yeah.
All right, let's rewind now and go back in time to sort of, what led you up to this moment. You started off as, I think you studied computer science undergrad. You were a software engineer at Google before you ended up in George Church's lab. I think most people like go to graduate school to end up at Google, not the other way around. So how did that like transition happen, and what made you go from very comfortable life in tech to being a grad student?
Yeah. So actually, it started just a little bit before the tech life. It was actually at the time when I was the TA for your class doing intro computer science, and then also intro artificial intelligence, where you might have been there too. At that time we were solving
backpropagation by hand.
Backpropagation by hand in Patrick Winston's class, but it was an important, I think, a very prescient kind of moment in both our lives. But I also had taken a bioinformatics class with Manolis Kellis at the time.
6047.
6047,
Yeah,
that's right.
I remember that class.
Yeah, and so that class was super cool. It was sort of, for me, it was actually, I actually took it as a sort of throwaway credit. I had one more credit. I needed something, and it looked interesting. But the moment I took that class, it was this amazing kind of revelatory moment for me, because it's for the first time I was able to sort of look and understand biology and look at evolution through the lens of next-gen DNA sequencing, and then looking for patterns in the sequencing, bringing algorithms to do that. And that for me was a sort of a huge turning point, just intellectually, of like, oh, wow, this is the kind of thing I want to apply myself to if I can.
So you took 604, the Intro to Comp Bio class, kind of like as a throwaway, but then you got the biology bug from it.
That's right. I'd always been interested in biology, but it always felt like almost on the boundary of like fiction in a way. Like I'd read these stories, or I'd read the sort of textbooks, and it'd be super interesting, but I didn't imagine myself as an engineer actually doing that, like memorizing things or sort of studying one little system, a very specific system it's great that there's folks studying that and creating this knowledge, but that just wasn't my nature as like a solve general classes of problems kind of person.
Sure. So you took this class, you got interested in biology at a deeper level. You go to Google, then what happens?
Yeah, so I already had sort of my momentum set towards Google. I was spending time working on a number of interesting problems and a number of less interesting problems. I'll come back to this, but my starter project at Google was to engineer a button, and when you press the button, you had data ingest from somewhere into somewhere else. So it's remarkable how much effort it is to code a button at a company of the scale of Google. So it was both simultaneously, I don't know if humbling is the right word, but educational is maybe how I'd put it.
Sure, you're like true cog in the machine.
Right. Yeah, exactly. But I learned a lot. I was learning a lot, but I was still very, very curious about biology. And a really fortuitous thing that had happened, and you'll start to see this as we kind of tell you about the journey. I'd met a person in the class, in Manolis's class, and who I worked with on a final project there. And that person was a guy named Adam Marblestone. He was my partner in that class, and we worked together, and I just really had a good mesh. He's working on DNA origami in some lab, and I learned a little bit about that. We had this productive project, so did the class. I went off to Google. He continued in his grad studies. About a year later, as I'm kind of in the middle of my about a year into Google, and I'm thinking about this is cool, but I just don't feel like it's like a little bit like I'm like a cog, a little bit too small for me. I'm like very hungry as a 23-year-old, like trying to figure out what I want to work on. And I'm thinking about sort of doing one of three moves. One is go to join a startup. Two is go start a company, and it'd be something more in like software world. I was quite interested in like knowledge management. Or a third bucket of other. And other encompassed various interests like biology. Although that felt like what would I actually do there? Do I go to grad school? What would that look like? And then there was this amazing moment. It was a like Tuesday night at midnight in California time, and I get a call from Adam Marvelstone. And I'm confident that this is a butt dial because Adam has no reason to call me at like 3 a.m. Boston time. So I don't answer the phone, and I'm like, that was random. But it was this weird moment of like, it just triggered a lot of memories of our projects together and of thinking about the biology. So for the next week, I can't like think about anything else. I just keep thinking about like, what if I could get in and just start doing biology?
You call him back?
So yeah, so like I reach out to Adam, say, Adam, can we talk? I'm curious what you do. I'm kind of curious about biology. And we hop on a call, and I'm like, Adam, just, tell me what you're up to right now. I'm curious. And Adam at this time, he's in his like second or third year of grad school, which most folks who have gone through a PhD know that that's like the nadir, where it's kind of like your first
sorrow.
Yeah, your first few projects probably didn't work, like nothing's working, and you're supposed to probably try to graduate in a couple of years. So he's kind of in that mode. But then I said, okay, Adam, it's all right. Just tell me, what are you actually working on? And he starts talking about like these like DNA origami, like nanotechnology formed from nucleic acids that fold on themselves. Starts talking about like doing like molecular recording, using DNA as using DNA as a molecular recording device for neurons. And so he's telling me about all these amazing ideas, and I'm like, Adam, that's amazing. That's amazing. I think I cheer him up during this time, and then he ends up having quite a prolific PhD. But anyway, I ended up saying, Adam, can you connect me with more folks in the lab? Like, maybe I can help some people. Like, I can code pretty well, that. Can I just be useful? So he's like, yeah, I think I can like see what I can do. He ends up blasting the lab at the time, and I get back emails from multiple people. So one of these people ends up being
like free Google engineer for everyone.
That was actually roughly the email.
That's
exactly.
pretty compelling. Yeah.
Yeah. And so people reach out. So I have people like Shri Kasuri, who's now the CEO of Octant, reaches out, and we talk about what he's up to. Mark LaJoy reaches out. He's now the CEO of Outpace. We talk about some of the like recoding, like genetic recoding projects he's working on. Dan Goodman, who's a professor now at University of Pennsylvania, he reaches out, and we talk more about this kind of work. And so I have these chats with folks, and I realize, holy cow, like these people are doing something really, really interesting, and just felt like I could work with them quite well. So I basically email back them and Adam and say, all right, guys, I'm down to show up. Like, what do I have to do? And they say, okay, George is going to be in San Francisco actually next week. Maybe you can go talk to him about that. So then, like, I don't know any better, so I like email George directly. They have me on a CC or BCC, and I just pop off, and I'm like, George, I would love to meet you. Can I, buy you coffee, etc. And so we schedule that, meet with George, have a great conversation, and I end up, funny enough, he's heading like from the city down to the Bay. I end up offering him a ride, so I like ride in the car and talk to him for another hour. Just have
that he accepted the ride. It means he probably wanted to spend time with you.
What's that?
It's a good thing he accepted the ride. It probably means he wanted to spend time with you.
It's actually funny. This is like a little bit of a taste of George Church's character. He actually asks me, he's like, by the way, are you driving anywhere generally in the direction of Mountain View? Like, can you drop me off as far as you can go, and then I'll make my way the rest of the way.
It's like do Uber and Lyft exist at this point?
This is like early Uber Lyft.
Okay, well, so George was like kind of hitchhiking with you.
Basically, yeah. He's like, "Can I hitchhike?" And I was like, "All right, George, I will drive you. I have time." So anyway, at the end of that drive, I would drop him off and say, "George, is it crazy if I just show up in your lab like in 2 weeks and just start working?" And he's like, "Well, I don't want to convince you to make a big move like that, but if you showed up, that would be great."
Wow. And quit your job and move to Cambridge?
Yeah, basically. So that was, and I actually just left Google like right before that to be precise. And the reason was exactly, I had basically kind of told my manager there that was like, I'm going to do one of those three things: start a company, join a company, or other might be something totally crazy like biology. And he said, that sounds great. Let me know in a month when you change your mind.
Were you like a PhD student in the Church Lab officially, or like, seems like you just like circumvented all of grad school admissions?
Not exactly. So I showed up. I gave myself a title of research engineer.
Okay.
On LinkedIn. I probably on paper I was technician, making technician salary.
Okay.
But I was happy to be there.
A little bit of a pay cut for
It was a bit of a pay cut. When you're 23 or 24, you don't really understand money, so that's fine. And you can do those crazy things. But yeah, just showed up and just started working with people and being helpful from day one.
That's amazing. What was it like being the computational software guy inside of the, academic environment of the Church Lab?
Yeah, it was interesting. There was the Church Lab world, and then there was the adjacent world.
Okay.
So
what's the distinction?
The adjacent world, so the Church Lab is embedded in the genetics department. And genetics department, a lot of labs are studying very specific genes and pathways. And so going to these talks, all you hear is these acronyms. And
a lot of acronyms in biology.
There's a lot of acronyms, and people are like talking about these different pathways and like experiments to deduce them. And it just never hooked me, because it felt like a one-off problem, like a very important one-off problem that would reveal something deep about evolution. But I couldn't quite, for me, what I was attracted to is what was happening in the Church Lab, which is almost the inverse. People were obsessed with building technologies that could answer hundreds or thousands of questions simultaneously, like basically trying to solve classes of problems through the building of technology. And so that was, for me, very educational. One is just to spend the time to understand the ways of thinking, and that takes a little while, but also to be introduced to this way of thinking about that's quite unique to biology, and the way you can engineer biology is you can do these things in a massively parallel way. So it's almost like the way evolution has shaped us, but now turned into an engineering discipline. And this is the sort of concept that you'll hear over and over about from people from the Church Lab of multiplexing, like doing many things simultaneously in a single system and getting huge leverage out of classically a complex experiment.
And what years were you in the Church Lab for?
So this was 2012 and until 2020 when we spun out Manifold.
Oh, wow. Okay. So you're there, but you ended up going from sort of research engineer to doing a PhD, right?
That's right. Yeah. The first 2 years were this research engineer. I was useful. So I was able to help publish a few papers that helped me get into the PhD program, which was the Harvard biophysics PhD program. And then even once I completed that, I was able to stick around and start thinking about applied aspects of what we do as part of the Wyss Institute, part of Harvard. And that would end up transitioning into what would become Manifold.
That's an amazing story. The Church Lab is definitely something special. At this point, I think dozens of biotechs have come out of the Church Lab, and I think it's very well documented. What do you think is so special about George and the environment he creates?
I think, yeah, it's the classic what's in the water question. I think there's sort of George personally, and then there's what the environment that he creates. So him personally, it's actually pretty simple. He's super, super smart, and he's super kind. And I think when you have the combination of those things in one person, it means that you get a lot of creativity and resonance. And you also get a lot of trust. So people really trust George. So even though he's involved in so many different things, people continue to come to him, share their ideas, and in turn get those ideas amplified and mixed and augmented. And I think that's super special. And then on the environment side, it's interesting because you have such a big lab and a decently well-resourced lab. On the one hand, you don't get a lot of George attention per person naturally. However, if you want to get George's attention, if you have a question, then he's absolutely there for you. And so it creates this selection pressure for people who have to if you kind of know what you want to do, you go out and ask for help to do it. And if you need the resources for it, whether it's to get the resources to do the study or some sort of collaboration, those resources are there. You just have to be clear about your ask and ask for it. And so it creates this actually training ground for future professors, future entrepreneurs, where you sort of learn to kind of be very proactive. What's that?
Advocate.
It's advocate and be proactive, but because the resources are there, you can get what you need, and then prove that you've sort of done well with that resources, and then be able to get more and more. And so that is, in a way, very parallel to how academia works, how entrepreneurship works. So it just organically turned into this amazing thing. And I guess the last thing I should highlight is because of, I think, George's kind of the culture you set up and the track record of the lab, you attract really good people there.
Sure, it's like a flywheel effect now where people know and they kind of self-select into it.
That's right.
Can you share any stories about his mentorship style?
Yeah, so George is a very captain of your own ship kind of mentor, and I've heard him use that phrase for folks. I remember, I think in a lab meeting, a grad student was complaining about like his other committee members were saying this and that and telling him to do this and that, and George just kind of had this one-liner of like, "You're the captain of your ship. What do you want to do? What do you think you should do?" And I think that style was very constant. I think there was a lot of this. Another kind of classic Georgism is he'd kind of say you do an experiment and you get it to go from like literally 0% to 1% working, and George would say, well, 1% is tantalizingly close to 100%. And that was like a very powerful idea. Like if you can get that 0 to 1, then it's effectively, you've turned it into, you've sort of made sure you've shown that it's possible. And now it's an engineering problem. So that kind of confidence building.
I like that. I'm using that
really
internally.
Good. So those kind of little kind of things. I think also, for better for worse, George says yes to a lot of things. That it's a little harder to do as an individual. And like you have to do, like especially starting a company, you have to learn how to say no to things. But being open, I think there's another George line I really like to use myself. It's think of it as an opportunity, not an obligation. And that attitude.
I get it. I mean, he said yes to you, right?
Yeah, exactly. Which was kind of wild. Like I was just kind of like, I could have shown up and not been useful or faded off. And so that was a great risk and sort of one.
Amazing. One thing I learned sort of like thinking through what to talk to you about is that there are 3 companies that came out of your lab bay alone, not just the church lab, but your bay. So Manifold, Dino, and Manifold Bio. And so you and your lab mates were all kind of, at least from the sort of looking back on history, it looks like you're all very early machine learning adopters in bio and founding kind of this first wave of AI first biotechs. Like what was in the water at the lab bay? And like what gave you all the kind of prescience to know this is where the industry was heading?
I think a big thing, this kind of, so for me, when I came to the church lab, I had this computer science and AI background, and I had this hypothesis that wasn't, the instinct was good, but the implementation was not correct, which was that I would come in with my knowledge of AI and machine learning and, bring the biologists out of the dark ages. Folks are doing experiments like pipetting, like I'll help them like automate stuff. They're analyzing their data with Excel or something, and I'll help them like code and do more kind of systematic analysis. That turned out not to be like, especially in the church lab, people were quite sophisticated with those pieces. But it turns out the nature of biology is that getting really good data and quality data and at scale is very, very hard. And what I realized is what people were doing in George Church Lab is building new ways to generate that kind of data at scales that were previously unprecedented, this like massively parallelized or multiplexed way of doing things. So looking back, what was actually happening, I think we're keenly aware, like very clearly aware that there's this huge exponential of artificial intelligence and machine learning. And we're at the like peak of this and, no end in sight.
Hopefully it's not the peak.
Yeah, exactly. It's the we're on the vertical part of the exponential.
Exactly.
But there was another exponential that was happening in the George Church's lab, especially, or the church lab had a lot of the people here, and this is the sort of the multiplexed libraries exponential, where folks were figuring out how do you combine DNA sequencing, DNA synthesis to turn biology experiments and protein engineering experiments into these sort of library multiplexed experiments.
Okay, so just general idea that, hey, like sequencing and synthesis, printing and reading are just like getting way cheaper. Like what can we do based on just writing that curve down?
Yeah, exactly. It's like if you can turn an experiment into a variant of a synthesis and sequencing experiment, now you just have the power of this massive parallelized, like,
sure, it's like
technologies that are getting
Moore's law
super cheap.
Computer science to work for you.
Exactly. Yeah. It's exactly the Moore's law. And in fact, like back then, like the sequencing costs were like
probably faster than
super Moore's law. Is like George Church would say it's like a hockey stick on a hockey stick in terms of drop. But it was this, but to be able to turn experiments into like this art, the dark art of multiplexing, as I sometimes like to refer to it, is actually there's a lot of tacit knowledge to that. There's a lot of like very much like apprenticeship / peer-to-peer knowledge. So that is what was happening in that bay. Is we were sitting in there with, we had like me and Eric Kelsick and Pierce Ogden and Serge Biswas from Nabla. We're sitting there. Dan Goodman was there as well. He's now a professor at UPenn. We had David Thompson, who was previously from David Liu's lab, so he was coming in mixing in some of those types of ways of thinking. We're all sitting there in one bay and just constantly helping each other debug experiments, brainstorm, try new things. And this was happening for like a 4-year period before, like, I don't know if any of us necessarily said we're going to like start a company, but we knew that we were building very, very powerful technology. And then like the AI piece was coming in where we were saying, okay, like this now fast forward to like 2016, 2017. And, deep learning is starting to really catch. And NeurIPS goes from this like niche conference to this like, starting to become this like big gathering of people trying to do these amazing things. And we start combining these two technologies. We start doing library experiments. We design a library, learn like patterns in that, design the next library. And this, Pierce and Eric are working on AAV capsids that goes to dino therapeutics. Pierce and I are starting to work on protein library experiments that becomes kind of, V-1 of what Manifold become. Serge is thinking about different kinds of proteins as well that will go into Nabla. So you have this just confluence, and also just like quite a long time of becoming deeply expert in these kind of two technologies and the confluence of these two exponentials.
So talk more about that V-1 of Manifold. Like, how did the idea first take shape, and like what was the original premise of the company? Because I'm guessing it's probably evolved a bit since you started.
Yeah. So when we started, the original idea was actually better ways to do antibody design discovery. So this was our hypothesis: is we would build the like huge library on like huge data sets of library and library interactions of lots of binders against lots of different proteins and the epitopes of those proteins. That would be the training set for building one of the like a true de novo binder model. And so we started designing all these experiments and generating all this data. We started trying to learn and train on this data. It wasn't just like working, let's say, that's like a pretty big hurdle, both from data gen and building the right models. And the other thing though is, even let's say we were making good progress, but we also started going out and talking to people out in the outside world. So folks in drug discovery, these are like director-ish level scientists in pharma companies. We're talking about to professors, to academics. And the feedback we kept getting, or what we kept saying is like, hey, we're going to build this amazing, here's our fancy way of making antibodies, and it's going to be super cool. The feedback we got is, well, binder design is kind of solved. Like, you can just make antibodies. We can go to a CRO, we can go to an Adamab, we have our internal teams.
Sure, and they'll run phage display for me or whatever. And
yeah, exactly. So people are like, that's not, it sounds like what you guys are doing is super fancy and cool, but like, how is it better? And we kept hearing that sort of feedback. And even the way we did binder discovery there's a lot of learnings that give us huge advantages today in Manifold, but it wasn't in itself a company, like as a sufficiently differentiated company. However, one thing we kept hearing from talking to folks was, well, that's not really a bottleneck. The real bottleneck is down the road. You get into these functional tests, and then you get into in vivo tests. Like, you still have to pick your best couple molecules and see which ones fly. And that's where the rubber hits the road and where things fall over.
Okay, so you were interested in like widening the funnel even from the early days.
So we spotted that, like, okay, that in vivo bottleneck is a really big thing, is reinforced by professionals in industry. It also happened to be that Pierce is working on viral capsid engineering for delivering gene therapies to specific tissues. Naturally, when you engineer a viral capsid, it has its own DNA inside of it, where this lab is one of the best at being able to do these kind of experiments that are libraries and DNA and barcodes and all that. And so Pierce is already thinking about in vivo screening, but for capsid for viruses. So this is when we start thinking, like, could we maybe unlock this for proteins? The challenge you have to solve for proteins, though, is DNA is not naturally present with an antibody, with a protein, like it is inside of a virus. So you have to devise a new strategy for barcoding if you want to unlock sort of the equivalent bottleneck widening technology for the world of proteins and antibodies.
And what was it like starting a company out of the church lab? Was it, did you just funding materialized for you?
I would definitely say funding did not just materialize. So one thing we benefited from, we were part of the Nucleate program in its first embodiment. It was literally 4 companies. And now it's this quite big program. It's a very competitive process. At the time, one of the founders of Nucleate, Sufyan, was coming to our bench and saying, guys, like, we put out these applications. No one's applied. Can you please apply? I think you guys would be good. We're thinking like, this is a waste of our time making decks or whatever. Like we're doing science. We're going to make this thing work. And then they will come. And this is like end of 2018, mid 2018, end of 2018. But we're having these conversations externally, and I'm appreciating the value of these things, sort of starting to do the equivalent of customer development.
Sure,
or
force you to tell the story.
Yeah, and so we realized, okay, I don't know, let's sign up for this thing and try it. So we sign up, we go through some iterations of building a deck, and I do think that starts to force some clarity of thinking on the product, the market, the business strategy, and facing reality where people are saying, that's not going to work, that's not going to work. But fast forward, we end up toward the end of the year realizing we have something quite special. Actually, one funny thing, at the end of Nucleate, we went and got invited to pitch some sort of angel group that Harvard had put together called the Alston Fund. And we showed up and just pitched this thing, and they call us the next day and they say, great, we want to give you guys like $50K, like uncapped safe. Or no, they said like $50K, and we're like, what terms? Like, you tell us, right? How about this like Y Combinator thing that we downloaded? And they said, sure, that sounds good. And then one of the other folks from that group calls us like, hey, I want to give you another $50K just direct for my fund. And I'm like, okay, same terms? He's like, sure.
That's great.
And so we basically, and then they say, okay, great. Like, you guys have a bank account, right? And we're like, so then we just don't respond for 3 days, go like incorporate the company, create like a bank account with Silicon Valley Bank, and then respond to the email. Yes, we have a bank account. So we put $100K in the bank. We have this like shell corporation of Manifold. We're still at Harvard, and you can sort of create a company as long as you're not spending your time or resources there. So it's just sitting there. And for the rest of the year, we're focused internally on research. But end of 2019 comes around, and then we start saying, okay, like we should probably start thinking about fundraise. Let's go start talking to investors. What do we need to do? 2020 rolls around, beginning of 2020. We're like talking to investors, like, okay, we're going to do XYZ experiment. And then what do we need to do for you to fund us? And then March 2020 hits and COVID hits. And so we're like, oh crap, we can't do our like experiments. And now we're just like sitting at home talking to investors. And in fact, a lot of those investor conversations, a lot of them are like, don't know what's going on either. They're like, I'm busy, or my kids are like over me. We're going to pause. So we're like sitting there thinking like, all right, like we can't get back in the Harvard lab. And most of the investors are like offline. Like, what are we doing? But a couple investors sort of stayed on it, I guess, during that weird time. So people think of COVID as like the beginning, like COVID as this easy time to raise, this, bubble time. The beginning of COVID is a very hard time to raise because most people are not sure what's going to happen to the world. But a few investors, were really, still staying true. So like 50 Years was one, just continued to be very active and was very interested in Manifold. And then Playground Global was another one we met. Jory Bell, who actually also was the seed investor for Strand, for Jake's company. So, these folks just saw what we were doing was quite special, and, were willing to give us a shot, and we were able to raise and get in the lab and get going.
Can you talk more about Pierce? Because it sounded like from what you were saying earlier that he was also kind of had one foot in the world of what eventually became Dino Therapeutics, Eric Kelsick's company. Like what was he just, was there some fork in the road with him where he had to pick?
Basically. So Pierce Ogden, he's my co-founder and the chief technology officer of Manifold. He's an absolutely brilliant inventor. So he is, they talk about the 10x engineers or whatnot in the sort of software world. Like, this guy's the
scientist.
He's the 10x scientist, right? Or the 100x scientist, the million x scientist, because we can multiplex. And Pierce was working with Eric on the capsid screening that would go into Dino. Pierce was working with me on the protein engineering that would be the foundation of Manifold. In fact, Pierce was the first author on the science paper that was the capsid screening and was the foundation for Dino. So it's barcoding all the way for all the things, and I think it was an interesting moment for Pierce where it's kind of like, Dino, Eric wrapped up, started going with Dino, and this choice for Pierce along the lines of, do I go off, in that direction, or do we kind of take the stab with this protein world? And what was very attractive about the protein world to both of us is it felt like a much bigger product space than the AAV gene therapy space. And I think that is really, really something that was attractive to us, to Pierce. Like with the protein space, you can have proteins conjugated to proteins, conjugated to oligos, conjugated to small molecules. So you have this like prolific space of potential medicines one can create, and just much more degrees of freedom.
Got it. So it's probably pretty appealing to someone who's got sort of an inventor at heart.
That's right. And also I think the challenge of, he had already solved the sort of DNA sequencing as a readout for AAV, but we were like very keen on this sort of concept of protein barcoding, like a peptide barcode. And that sort of idea had emerged, and Pierce had, come up with it, and it was like thinking about how do we like build this? How do we come up with this, make this real? And this is what was floating around in sort of, a really compelling, I think, scientific challenge, in addition to a big opportunity. And, we were both really excited. How do we reduce this to practice and make it real?
Totally. You have a third co-founder, Shane Lofgren, who joined as the head of BD. I would say especially back then when I was looking at kind of biotechs coming out of sort of academic labs and so forth, having a BD person on the founding team isn't super common. Why was it such a critical strategy for Manifold?
Yeah, so Shane, his title at the beginning was head of BD, and in practice, that's probably what the sum of activities that he did could be called. But what Shane really was really pivotal on is becoming our first person really thinking deeply about product. And I think this is a really, really key thing to what's been successful, has made Manifold successful from a company build perspective.
Mm-hmm. How'd you meet Shane, by the way?
So I met Shane at a tea party.
What?
A tea party, which was hosted by a church lab, a fellow church lab member, Dima. So he invited a number of his folks that he had met in Boston, as well as other church lab people. Dima had met Shane because Shane had moved to Boston and was joining some of the Atlas Venture runs. Mm-hmm. And this is like this run that they do on in the mornings. So he had met Dima. I met Shane at this tea party, and I just immediately recognized this guy as incredibly generative and creative, and also had been sort of hanging out with the Atlas folks. He had started, helped. He was previously, before that, he was a bioinformatician in a lab at Stanford. Mm-hmm. He had then followed one of the postdocs to MD Anderson, and then continued to work with him there as a bioinformatician. But he also worked in the Stanford OTD, helping spin out companies. And so back in Boston, he had come to Atlas and drawn their attention to something coming out of the lab he was coming from. So they had actually created a company out of this. It was a small company that got rolled into another company called Icana Oncology. And so he was quite talented at both. Was very technical, which is important, but also had some reps about thinking about sort of how do you actually create a medicine out of a platform technology. So I met Shane at this tea party. We'd gotten to know each other over the next year or two. And just this funny story where, Pierce and I get going on starting to making decks for Manifold, or there's all this platform stuff. The product thinking was like very underbaked. I mean, we just hadn't really developed those neurons. It's something that's tricky to do. And I remember sending Shane our deck, and Shane is very, very verbose in his writing. He wrote me back a 2,000-word email that was basically just
pre-ChatGPT 2,000.
Pre-ChatGPT,
2,000 word email.
Exactly. Okay. Yeah, you can imagine what Shane plus ChatGPT are like these days. It's an unstoppable force.
About like what you could do better in your deck, or
yeah. Well, he basically was like, "This is cool. I kind of get the technology." He didn't fully get it yet. The product the applications are really underbaked. Like, well, why? What is the connection between your platform and your product, and like what you're trying to do? Like, you guys can do this in vivo screening thing, cool. And also, he didn't quite dawn on him how powerful it was, what we were claiming we could do. But also, he was saying, it's not clear. Like, you're like kind of saying you're going to go after cancer, but like, what problem are you solving for different types of cancer? So then he wrote this giant email criticizing it, and at the end, I wrote back like a one-liner that was like, "Well, Shane, what do you think we should do?" And a day later, I get this like giant email back that's like, well, actually, the more I think about it, blah, 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 here's like your app advantage. And he starts thinking about these ideas about targeted medicines and like solving the targeting problem. And at the end of the email, he says, like, this is pretty exciting. Can we meet and brainstorm? And so we meet, we brainstorm. We also explain to him how the technology, like at least the idea for how the technology will work. And it kind of blows him away, like just the idea of how we're going to like crack this puzzle of protein multiplexing that no one else has been able to crack. And sort of the rest is history. So fast forward, he starts playing this role of like going out and talking to people, really mapping out the product directions, doing the equivalent of customer development. And fast forward, he's running strategy and portfolio strategy. And what BD is today is a little different. BD today is more like transactions, et cetera, like actually doing the deal. But I think that product or customer development piece is really, really key. It was key then, is really key today.
Do you think every biotech needs someone on the founding team thinking about product development?
Yes. Like I think that is one underestimated thing, especially for platform biotechs, where if you truly think you have a lot of opportunities, you really need to start thinking about which ones, how do you stage the order in which you do that, what matches your what matches your strategy, what's going on in the outside world, and what you need to work on first.
Yeah. Let's come back to platforms, but you've sort of described Manifold as having been through the idea maze, obviously like COVID, complicated things. What were like the dead ends or hard turns that really clarified your focus on platform, product, and even the indications to go after?
Yep. Yeah, it's the idea maze. I got that concept.
It's a biology concept, I think.
Who is it?
Biology.
Oh, okay.
Yeah.
I got Sri Kasuri kept saying it back when we were like thinking about starting the company, and he was like in his own idea maze at the time, and he kept using that phrase. But it's a good one because you're trying to figure that out early days. And for us, there's a number of factors. One is, what is your technology good at? Two is, what could possibly be funded? Three is kind of like where is science and like clinical medicine today? Like these are all factors that play into this. And at the time, this is 2020. So, we had basically been able to articulate, we think getting medicines to go to a specific place in the body is a good focus. But then we had everything. We had, oncology in the sense of getting drugs to tumor and avoiding healthy tissue. We had, we did have brain, like penetrating the blood-brain barrier as like a problem to solve. But the ordering of those things we had was that in 2020, people were still very nervous about neuroscience. There's a lot of failures.
Dead in pharma, basically.
Exactly. Yeah. Like not a lot of investment. So that we're like, okay, it's on the blue skies slide in the deck.
Oh, so you were actually like pretty broad in the early days. We're like, I don't want to say unfocused, but you had a lot of...
Well, yeah, like we were telling a big story of like, if this works, like here's what we can do. But we did have a sequence that we like when we raised the seed, we said, okay, today we're going to work on oncology, and specifically we're going to focus on getting cytotoxic payloads or like T-cell engagers to go to specific tumor types, either both liquid tumors, and then we got attracted to solid tumors because that seemed to be a little more untapped area.
And you eventually though had to kind of like pivot or evolve the company from T-cell engagers to the blood-brain barrier, which was originally, you said on the blue sky slide. That seems like a big bet the company situation. Like talk to us about that leap.
Yeah. So like the first couple of years of the company, partly, the value of a good product a good product definition is it creates the pull for your platform. So having T-cell engagers as the goal was still a really good product pull. And it actually made sense from where the market was. So until maybe 2022, mid 23, we're focusing on that as the application. We're the drugs we were testing and barcoding and putting in vivo were T-cell engagers. But then mid 23, two things started happening. One is, in pharma conversations and even as early as JPM 23, a lot of folks were asking us about brain shuttling. Can you do brain delivery? And we realized like scientifically that was starting to become a little more aware, or people were becoming aware of that. The other thing that was happening is some of these neuroclinical trials were showing some interesting signal. So some of these Alzheimer's drugs in particular, like Leqembi out of Biogen or lecanemab was starting to show interesting data, and people were believing it's probably going to get approved. Donanemab from Lilly is starting to show interesting data. So all of a sudden this starts to create this interest in brain targets again, and people appreciating, okay, like this could be really valuable. And these are massive indications when you go after brain indications. Like Alzheimer's is millions of patients in the US, hundreds of thousands being diagnosed every year. The other thing that happens is we start doing our own sort of pilot studies internally on brain shuttling. We've sort of hit this like about 100-plex scale now in vivo, and we're able to go. And I should highlight that in year 1 we were like literally like 5-plex. Year 2 we were like 10-plex. Now we're at like 100-plex. So now we're starting to do these brain shuttling studies where we've raised candidates, antibodies that can go against a specific receptor and cross the blood-brain barrier. We're now seeing interesting data in these screens. And we think we're like, oh, wow, I think we're like really positioned to take on this challenge. And then this really important moment happens in about November of 23. Roche published this amazing clinical data of a brain shuttle attached to an anti-amyloid beta antibody. In fact, it was a failed anti-amyloid beta antibody, gantenerumab. They put a brain shuttle on it. This is a molecule that facilitates crossing the blood-brain barrier. And they show that they have this remarkable fast clearance of plaques in Alzheimer's patients, and it does away with some of the symptoms, including this brain hemorrhaging type phenomenon, ARIA. It's amyloid-related imaging abnormalities. That data was the shot heard around the world. Like we saw that data and we were like, holy cow! Like brain shuttling is now a real technology. It's not just a brain shuttle is nice to have on your medicine, but any biologic to the brain is going to need a brain shuttle to be a good drug. Pharma, we started to see recognizing this as well. I think people's jaws dropped. Like multiple times I heard people saying, my jaws dropped when I saw that data. At the same time, oncology world is starting to become, from a funding perspective, like one, the funding market is in general decaying. Second, the sort of the barrier, or let's say the bar for oncology data is increasing. It's becoming more and more competitive. So you really are facing having to raise a ton of money that can fund you to a pretty far clinical readout, and really starts forcing the company towards like really betting the farm on like an asset or two. Mm-hmm. And so those forces were kind of like making us wary of going of falling into that sort of path for oncology. Meanwhile, the brain direction is pulling us, and it also feels like a really nice fit for our platform, potentially even better in a first order than oncology. Because like in brain, like ultimately we are this like multiplexing, like PK multiplexing engine, like as a first order. And the two of the defining properties of a drug are its PK and its PD. So if you can pick a problem where PK and PD are really closely tied together, so the PD being like the efficacy of the activity of the drug in the brain, that would be a really, really strong fit for our platform. So we kind of went to the board and we said, we should really double down in this brain direction. We see pharma pull and interest. That's going to be really good off-ramps for partnering. Oncology is a bit of a hairy space right now from a funding perspective in the bar. Like this is the commitment we want to make, and we shifted the company, and like I would call it a pivot in a sense, or at least a focusing event. That's end of 23 and beginning of 24 turned out to be a very good decision.
So now you're fresh off a brand new collaboration with Roche. I read about it in the news. It's a $50 million upfront, which in these times is pretty unusual. So congratulations. Your bet seems to be paying off, and you're collaborating with them on next generation blood-brain barrier shuttles for brain-targeted drugs, so a problem that's challenged the industry for decades. Can you tell us more about the partnership with Roche and what you're trying to achieve?
Yeah, so it's a super exciting partnership for us. It's the biggest AI bio platform deal that's happened in the last few years in this fairly quiet time in the industry. I'll correct you, it was a $55 million upfront.
Sorry. I was counting.
Who's counting? $2 billion and over 2 billion milestones, significant royalties. There's even a possibility of participating further in co-funding development and getting more royalties. So this is a fantastic deal for us.
Wow. How long did it take to set up the partnership?
We've known the Roche guys since we started, and literally we've been talking to them since we started the company. And I think that's something that's, not to go underestimated, is showing people a trajectory over time is the best way to build conviction. But the important thing that happened is, as they themselves were getting excited about their own brain shuttle work, that was that kind of fall of 23 trontinemab data, they started to build conviction about that space even more broadly. And they're thinking about what's the next generation. And this is the team that's positioned to recognize and understand what are the hard problems, where are the opportunities. And so Manifold's continue to build our in vivo multiplexing platform. We keep checking in every once in a while. And then we show up and say, hey, this quarter, we're showing you our platform applied to brain shuttling. And they're like, whoa, like that's super cool, and very, very interesting for a few reasons. So we start having these meetings, and it's still probably a year of kind of getting to understand what we're doing and showing momentum there. But the big thing that the deal is about is going after novel receptors, or not, we call them portals, novel portals into the brain. So Roche has unlocked their brain shuttle is transferrin receptor. Most of the world has been working on transferrin receptor for brain shuttling. There's sort of some one-off instances of using new receptors. Transferrin receptor is it's the devil. It's got very good properties for certain applications, but there are certain limitations. It's very broadly expressed. It's expressed in certain cell types and not expressed in others. So if you start thinking in a more refined way about the every single different brain product, you realize that a different profile would be desirable, more specific to the brain or expressed on certain types of neurons. If you want to deliver not an antibody but an oligo, then you want to be able to internalize into certain cell types. So there's all these gets much more of a rich product space when you stare at it. Roche understands this, and we understood this, and we were already building towards it, really unlocking new receptors. So the deal is structured that we are creating shuttles to new receptors. We do it with our in vivo way, where we can test thousands of antibodies against every different receptor and just empirically figure it out in vivo. And then we're just licensing rights to a specific handful of named programs to them. So we get to keep our shuttles, we get to evolve the shuttles and make them better. We get to use them for our own programs. We get to use them for potentially other partnerships. Roche gets, together we're going to build potentially some of the next great medicines that have not been possible before. So it's a fantastic synergy of forces, and just super excited about this.
That sounds amazing. Let's talk about platform companies.
Yeah.
The term gets used a lot in biotech. Platform companies were very sexy and in vogue a while back, now less so. How do you define a true platform company, and what do you think separates a sort of real platform company?
Yeah, so my view is the platformness of a company is defined by the breadth of the product space. How many products can you really create out of that? And so there's I like to kind of say there's lowercase p platforms and uppercase p platforms. So lowercase p platform might be like a type of like an architecture, like a type of T-cell engager or something like that. Mm-hmm. Even one of our shuttles in itself could be considered a platform. It's opens up maybe targeting to the brain. That's a few brain programs, or one of our shuttles to other tissues can be an adipose targeting platform. But what an uppercase platform is, uppercase P platform, is something that really just has a prolific space. The ultimate platform back in the day, for example, was recombinant DNA as a industrialized technology. All of a sudden, you just opened up all of these both natural proteins, eventually engineered proteins like antibodies. That was mega uppercase P platform. Where Manifold lives is where we have this huge opportunity to be able to drive drugs to go to specific tissues, and this is a very, very big problem. Today, what's been solved is effectively delivery to liver. It's sort of the natural thing. Mm-hmm. But beyond liver, extrahepatic delivery is a very, very hard problem, especially for all sorts of modalities that folks are interested in doing that for oligos, for certain kinds of peptides and proteins.
So is your advice to entrepreneurs, if you're going to build a platform, go for a big P? Don't build too small of a platform?
Well, there's value to be created in all sorts of ways. I think the font size of your P is what defines the strategy of the business that you can build. meaning like how much partnering you can use in your strategy versus how much you have to, kind of reserve rights and maybe bet sooner on a few specific assets.
I see. It does feel like the sort of like golden era of platform biotechs was, like Genentech, you mentioned, recombinant DNA and Regeneron as an example was a long time ago. And today the emphasis is really on product over platform. It feels like investors today are kind of wary to pay for the platform and just want the drug. Like, what do you think is going to make the pendulum swing back in the other direction?
Yeah, it's definitely a challenge today. The funding environment's harder. The platform companies typically, I'd say, have a longer J curve that you dip down and you come back up. It's actually interesting from like and today a lot of you just need more capital up front, and also you need to kind of lay that foundation of how you are going to sort of walk that arc if you're going to try to build a platform company. It's just, again, something that's very tough in this biotech environment, although it seems like maybe we're coming out a little bit. And so we actually, one thing that was interesting that we brought on at Manifold, the chair of our board is a person named Steve Holtzman. So Steve Holtzman has written about this sort of platform product strategy dilemmas over the many years. Originally, he was the CBO of Millennium Pharmaceuticals. That was like also one of these pretty much like uppercase P platform companies where they were industrializing gene sequencing and licensing rights to different genes, and were able to do a ton of deals of non-dilutive capital. But he likes to say that ultimately to build a therapeutics company in the limit, you need a lot of capital to build those medicines. You can either get that capital through raising equity at favorable terms or through partnerships. And as long as you don't do too many partnerships that give away too many rights such that you have enough to grow the big value of the company, then you should be able to pull this off. And so what's for this kind of company to work today or in any time, you really do have to have a lot of substrate for partnerships, and your substrate has to be super differentiated and able to speak to acute strategic needs when they arise. So Manifold story, brain shuttling and brain delivery comes up as a really acute active need. We're positioned to really seize on that momentum and build out a prolific brain franchise. But it's still something that investors have to get comfortable with, that like, hey, I'm not going to just not necessarily do a speed run to a phase 2. Like, I'm going to lay the foundation here. But if we're right, we're going to have a much bigger opportunity in the limit. And actually, one thing that just reminds me of this conversation with Steve is we were just talking about, as we think about, who the next investors would be for Manifold's round, et cetera. And, these, you've probably heard in these words in the world, there's this sort of like one extreme of like founder-led bio companies, and then there's like trad bio, trad investors. And we were saying this to Steve, like, I don't think, the trad bio investors not yet comfortable with true uppercase P platform companies. And he's like sitting there scratching his head, and he's like, "What is this trad bio term?"
Kids.
Yeah, because we're like, "Well, what do you mean?" It's like, it's like single, like low number of asset companies. And he's like, "Okay, you mean product companies?" We're like, "Sure." And he's like, in my book, the trad bio or traditional biotech is Genentech, it's Regeneron, it's Amgen. It's the companies that built these platforms. The original biotech companies were big platform companies, and so it's funny that's kind of come full circle in a way.
That's so good. Do you think like AI changes anything for platform companies?
I think AI increases the pace at which these things can happen. Like, AI can now generate ideas, both in terms of composition of matter, but also in terms of product space. And of course, you're working in synergy with the AI. Like for the ideas, you have to really stress test them, and then that gives you a clue where to go. But then you have to really build the case. On the composition side of things, you have to have an advantage of, one, the data to generate and kind of guide the AI, but also even test what the outputs of the AI are. But nonetheless, it's definitely accelerating things.
It's pretty hard to hire folks with machine learning AI backgrounds today. What's your how do you fill those roles, and how do you compete for talent?
I think it comes down to that idea where folks have to be very passionate about the substrate that they're doing the AI and ML on. Like, there's a lot of exciting things you can do with AI today. It's kind of changing everything, but being able to like manipulate like atoms in like a chemical context is super, super exciting. And knowing that if you do that right, you're going to actually make a medicine that's going to be very going to be able to not just treat a patient, or not just like help a patient, but potentially even cure them of something.
How are you screening for that?
I think we just look for curiosity about the biology, and it's almost self-selective. You like, especially with like, you know, the reality is like the salaries people can get just doing, if you're sort of a full AI mercenary, you don't care what problem you're working on, or if you're like one of the lead in the world of like pushing the foundation models, like, you know, we're not going to outcompete OpenAI on salary, unfortunately.
Not yet.
Maybe the value of our equity. But nonetheless, you get people who are really excited about that, the domain, and also the idea that biology is very, very unique in that it's a place where you can create hypotheses with your AI algorithms, and then you can go and test them like at the scale of hundreds of thousands. There's not really like another like such like perturbative domain, as perturbative of a domain, or maybe not as perturbative in like a kind of like a good way. So maybe some weird experiments are happening on with like social media and stuff.
Yeah.
But it's just, I think that's a very addicting cycle. And then knowing that you're accessing data that no one else has in the world, I think that is also very, very addicting. So there's those pieces are common to our AI scientists.
It's not easy to find people with formal training in both kind of AI and biology. How do you think about like bridging that gap, and do you advise others to recruit from tech or sort of upskill existing scientists?
Yeah, you know, it's
a lot of people in tech would not be willing to quit their jobs, take a 90% pay cut, go work as a tech at the church lab for 2 years.
Yeah, and it's very real. I mean, it's funny, over the years I've been introduced to like, you know, dozens of people where, you know, a friend of a friend says, "Hey, talk to my friend. They're disillusioned by building, you know, whatever like social media app. They want to like do biology. They think it's super cool. Can you tell them about your transition?" And I'm like, "Oh yeah."
Just send them your W-2 from the year after you Google.
Well, yeah. I mean, I mentioned that, but I'm kind of like, well, I spent 8 years in the academic world, understanding and like learning the craft here. Like, you know, the first couple of years was just being helpful and working with graduate students. The next few years were as a graduate student myself, sort of wrestling through problems. And as I describe this path, I see a lot of people die inside because they realize this is not the quick fix to their like existential boredom. But I've seen a few people have done it, and they leaned in and they started really learning and understanding the domain, and just getting in there and doing stuff. And in the ways that the people that I've seen succeed, most of them jumped into like a lab and worked a little bit with a lab. A few have gone directly into a company, although I haven't seen the sort of direct to company path stick as well. I think like you have to like get really into the science, I think, to really kind of bridge that gap.
So Manifold recently released Ember, which is a fully open AI tool for de novo design of epitope-specific antibodies. To validate, you ran the largest de novo antibody experiment to date, greater than 1 million designs tested and measuring over 100 million interactions. That's huge. Tell us why you made it open source.
Yeah, we created Ember to solve a very specific problem that we had, which is all of a sudden we found ourselves with measurement capacity in the millions, now millions per month. And what are we going to put in that capacity? Even like doing phage display and yeast display, which we're very good at, was not generating enough quality hits to sort of fill up the capacity. So created this extra forcing function of like, how do we make designs that we can put in there? And about a couple years ago, the kind of diffusion-based binder design revolution hit with the RF diffusion work out of David Baker's lab. And then some really interesting work came out of sort of the Bindcraft world, Sergey and Martin and their students. And we started looking at that and realizing, like if we could kind of tame that and use that to specifically solve the last gap, which was making it work for antibody type formats, that would be very powerful. And we were kind of like surprised that no one had really done it yet. It felt like...
Yeah, why do you think?
So one thing we were thinking, like, well, maybe it doesn't work. Like there's some reason it doesn't work. So we, nonetheless, we tried it. We said, okay, like let's do certain, try a few strategies to get these methods to be able to work for antibody-based design. Some structured templating, conditioning using language models of how we select the sequences before we try, test folding them effectively. And we tried a few different parameters. And one thing we were able to do is we tried a bunch of different settings and did experiments to see which ones are actually giving us fruitful binders. And we found a set of settings that was working quite well. Mm-hmm. And so we realized, holy cow, we have de novo antibody design, or in that case, specifically VHH design, which is the format that we really focus on. Mm-hmm. And we realized wow, this is a huge edge. But we started thinking about others are going to inevitably figure this out. And it's only going to be a moat for so long in itself. And then Chai comes out with their closed model, but saying, hey, we can now do de novo antibody design quite well. Nabla announced they can do de novo design. And we're sitting there thinking, well, what's special about Manifold? It's the data. We have this amazing model, but the data is really what makes what we do really special. And we want people to know that we are doing really interesting AI work. We want to kind of draw attention to us. And this is built on open source work to some extent. So why don't we open it up? And it's a statement about what we think is important, which is the data moat. We're not opening up, we're starting to learn from this data and now training a model based on this data. That's a little bit of a different question. But nonetheless, it was a chance to say, hey, let's get this out there. At the same time we launched it, though, there's another really cool paper out of ARC Institute called Germinal, and they came out a few days before us, and we were like, okay, their method is pretty similar to ours, so our intuition that this is going to be figured out was correct. But nonetheless, it was a good chance to say, plant a flag and say, we're one of the doing some of the most innovative AI work, where maybe folks didn't fully appreciate that we're not just doing in vivo multiplexing.
I think more open source in bio is awesome. I'm here for it. Do you think there's gonna be more open source in bio? What's the next bottleneck to get more usage of it?
Yeah, it's interesting. It's there's open source models, there's open sourcing data, and then there's always going to be this question of value capture, and how do you?
Which I assume is why people do not open source as much today as maybe other places like tech.
That's right. Yeah, and even we've seen this phenomenon. One thing that what happens though is when something gets open source, it kind of catalyzes something else getting open sourced. AlphaFold was closed and until was more closed, then OpenFold started appearing, and then AlphaFold started to get more open, or at least following with releasing. So there's this nice just kind of benefits, and it the mode erodes, and now you kind of switch from okay, I have my advantage to let me sort of capitalize on sort of sharing what I have and drawing people's attention to what I have is really powerful. But I think there's gonna be a lot of interesting dynamics over the coming years about companies choosing to open data versus close, versus sharing, versus figuring out models to kind of capture value off that you're creating, especially for companies.
Yeah, I think the business model of sort of model companies is going to be very interesting to watch unfold.
Yeah, these are big open questions. If you're just creating the molecule or just doing the design, these are hard questions to answer.
Yeah. Whenever a new technology like AI takes off, there's obviously a lot of debate about whether it's overhyped. We're obviously in a very exciting moment for AI right now. What do you think is tangibly different in biotech now versus a year or two ago?
I think a couple of things. One, the AI technologies are more powerful than they've ever been. The ability to design a binder from scratch that can bind with tens of percent, with 10 or greater percent accuracy is insane. That's, or some people are reporting even higher. When we were thinking about starting Manifold, kind of V-1 of Manifold was building the data set to build something like this. It turned out the space of solved protein structures and sequence alignments actually had the information to facilitate this already. But the other thing that's changing now, in addition to the powerful models, is the data. There's so much rich data being generated that's very complex and very bespoke in its own different ways. There's very complex, the kind of data Manifold generates with in vivo. There's all sorts of complex mass spec data on kind of interrogating structures at a very small scale and interactions of different kinds of types of molecules. There's all sorts of this just the single cell data, the spatial data. there's all this like really rich data that's coming out, and then like immediately that data becomes useful to iterate and train on top of models. And so like the pace of all of this is really, really different than it was even 5 years ago. And I think we're going to see we're already seeing things that would have looked like magic a year ago.
Any predictions of what we see 12 months from now or 5 years from now?
12 months from now, I think we're going to see that, like, we're going to think of antibody design in terms of like getting a binder to like any epitope as like
solved problem,
like basically solved. Like I can get like roughly an antibody to roughly an epitope, and like maybe even like be able to vary the affinity, and it has like pretty good properties. I mean, we basically effectively just saw that recently with a few of the releases, and we're doing that internally as well. 5 years from now, I think we're going to be taking a molecule that's basically ready for prime time directly to a patient, potentially. I don't think we're going to be quite doing zero-shot clinical design, like literally taking that molecule straight to a patient yet in 5 years. But I think you'll see molecules that effectively are basically good enough for that. Because, and part of that I think about Manifold, we already have molecules that we put in vivo and they look really, really good already. So
you're saying like first in human in less than 12 months kind of thing.
Yeah, exactly. And just like, you have to evolve the confidence there and make sure that we feel that the data is like we really feel that that's gonna be true. Like maybe there's always gonna be a little bit of like testing right before, but I think in spirit of it, I think that the gap between design to going to a patient is going to be very narrow. Now, how do you get there? It's gonna be true for some applications. I don't think this is gonna be true for all applications. I think where there's gonna be a lot of edge is who can, like, for given applications, build the data, I'll call it the data bridge, to go from kind of like the best AI capabilities to the AI that can actually design that molecule for that application. Like if it's like an oligo that goes to like a specific tissue, specific cell type, you might have to build a little bit of like a path for the AI to follow to get there. Or if it's like engineering, much more complex enzymes that like can really maybe along the lines of like these genome editing, base editing type technologies, but like even more precise, more dialed in, more specific.
That's exciting. Any advice to founders who are getting started today?
I think definitely take seriously getting the domain knowledge of whatever problem you want to get into. It's one thing to come and throw AI at the wall, so to speak. But as we all have seen, like whenever you use AI and like something you're deeply an expert in, it's constantly this like uncanny valley experience because you can see the
flaws,
the flaws, and like that's the case. So whenever you're using AI for something that you're like aren't familiar with, and you're like, wow, this feels like it works, it probably doesn't exactly. Like it might be good enough, but getting deeply expert in the domain, that's going to be the key that keeps you like inventing that sort of key enabling piece to go from uncanny valley to like really useful, or like take the creativity, the output of the AI, and then you sort of take a human will probably still be doing the last mile for a number of things. And that last mile is like really, really, really valuable and often incredibly hard.
So be a domain expert. Makes sense.
Yeah. Pick your domain.
All right. Let's have some fun. Lightning round.
Okay.
All right. Say the first thing that comes to mind. You don't have to explain much. Just a few words. No wrong answers. First question, in your opinion, most game-changing medicine of the last 100 years?
Not a single medicine, but I think recombinant DNA as a platform is kind of mind-blowing because we entered the sort of digital world of drugs. It's like print a sequence or take a sequence from nature, create it, becomes a drug.
All right, I'll accept that as an answer. You can wave your magic wand and get a cure for anything. What would it be?
Alzheimer's. I think that's something that affects so many people that would be absolutely, as we push more diseases out, like that's going to be one that if we can solve will really increase the life quality.
What's something you were obsessed with as a child?
I was obsessed with skiing and jumping on the trampoline to get better at the tricks for skiing.
Oh wow. Okay.
So it was like a summer jumping the trampoline, learning all the little spins and flips.
Oh, and so you're really good at skiing.
I was a pretty hardcore. Pierce, my co-founders, we share that. We're pretty intense skiers.
Oh wow.
And he's a snowboarder.
Which dorm did you live at MIT?
So freshman year, I lived at Next House all the way at the very far end.
Me too.
Okay.
Freshman year.
Okay. And then I moved into a fraternity over on the Boston side called Phi Sigma Kappa.
You have 2 skill endorsements on your LinkedIn for making guacamole. What's
Wow.
your pro tip?
Is that more skill endorsements than my Python skills? So people have told me my guacamole is fierce, which I'll accept. A lot of lime and a lot of onion.
Okay.
More than you think.
More than you think, and then still some. Who should we interview next?
A couple people that come to mind. Let's say, if you're interviewing academics, I would really consider Dan Goodman. So I worked with him for many years. Just absolutely brilliant guy. He's a professor at University of Pennsylvania, a fellow Church Lab alum. On the founder side, it's a really interesting company called Vant AI. They've been pretty quiet to some extent. They've had some models. Their CTO, Luca Naif, is a super interesting guy and has thought really deeply about this intersection of AI and data generation. I think the audience here would learn a lot from hearing from him.
Cool. Thanks for the tips. Gleb, thanks so much for joining us on Transcribed.
Great. Yeah, thanks so much. It's been a pleasure being here.